Hello everyone. So this is the biology final practice quiz on the AP Classroom. So I'm gonna split this into two. Okay, I'm going to do one video for the MCQ since they're so short, they're so easy. Most of them are repeated. Okay, and then one for the FRQ, which there are a lot of FRQs. Okay, and there are a couple of hard ones. I'll see if I'll explain all of them together or I'll just explain the easy ones and then I'll keep the hard ones until I understand them. بس يعني I'll see how it is. For now, I'm going to do the MCQs, so let's just start. This came in the last periodic, so as you can see here, we have hydrogen moving from an area of low concentration to an area of high concentration. To go from area of low to high, it's called active transport, and for active transport, you need ATP. If we we need ATP. So then they ask us to which of the following best predicts the effect of not having ATP. So if we remove the ATP, what's going to happen? The H plus ions will stop moving through the protein. Now you're going to tell me, why don't the H ions start to move from an area of high to low? Why don't they start diffusing? Now you don't know if they'll actually start diffusing or not. You can't guarantee it. Okay, they didn't tell us that it can. They're telling us with ATP, the H plus will go from low to high. Without ATP, it will stop moving. Okay, here they're like conducting an experiment and they want to understand whether a new found membrane protein is involved in membrane transport of a certain particle. Okay, they want to see how they can prove this. They wanna do an experiment and see if this protein is responsible for the movement of the materials to the plasma membrane or not. The way that they can test this is they put more of the proteins and then they measure the rate of the particle movement. If the measure of the particle movement increases, معناته yes, it is responsible. If it doesn't, معناته it's not responsible. Okay, so you just add more and measure the particle movement. Okay, so here we have a CFTR protein. Which of the following best describes how the amino acid deletion will affect the structure of the CFTR protein? Okay, so where are we removing the amino acids from? The CFTR protein is made up of 1480 amino acids linked together in a chain. Some humans produce a version of CFTR protein in which phenomena an amino acid has been deleted from position 508 of the amino acid chain. Okay, so we deleted an amino acid. What do you think is going to happen? It will affect the primary, the secondary, and the tertiary structure. Okay, because if you remove an amino acid, that means the primary will get messed with, and then if the primary is messed with, that means the secondary will be messed with. That means the tertiary will also be messed with. Okay, so if you mess up with the primary, which is the thing that has the amino acid chains linked together, you'll mess with all of them. Okay. 6. Which of the following amino acid components shown in figure 1 best determines the interaction between an embedded protein and the hydrophobic regions of the cell membrane? So the thing that makes the amino acids hydrophobic or hydrophilic is this. It's the R group. Okay, so the thing that's involved with the hydrophobic or the hydrophobic embedding, the embedding of the protein is the actual R group. So the R group bonded to the central atom. So the R group, the variable group. Okay, here they're asking us for the endosymbiotic theory. It's because the organelle has a double membrane. That's why they say it's engulfed. Because when something eats, if I had this and then I had this big thing, and then this actually started and then this decided that okay, I'm gonna eat this, then it will have two membranes, one membrane here, and then the second membrane is that one. So two membranes means endosymbiotic event. Endosymbiotic theory is backed up. That's what backs it up. Which statement best describes the effect on water transport across the cell membrane if the aquaporin in the figure cases to function? Okay, so what are aquaporins? Aquaporins are they're proteins, okay, that help move water in in bulk, big amounts of water. Okay, water can still move freely through the plasma membrane. Okay, it can still diffuse through by osmosis, but it doesn't diffuse as fast. So this aquaporin, if we remove it, it will still diffuse but not as fast. Okay, so water molecules will still be able to move across the cell membrane but at a slower rate. Which of the following best describes what will happen to the movement of ions if there is a change in the cysteine of ATPase? Okay, so let's read. ATPase requires the use of ATP to move ions from one side of cell membrane to another. The amino acid cysteine is important to the function of ATPase. So this amino acid that's called cysteine is important to the function of the ATPase. A change in the cysteine prevents the ATPase from functioning. Okay, so now the ATPase stopped functioning. That means the gradient that the ions are moving in will all become messed up. Okay, like the gradient that it's moving in is, let's say, from a high concentration to a low concentration. Okay, we stop moving the ATPase. We mess with the ATPase, and then we mess with the cysteine amino acid in the ATPase. That means that the ions will stop moving, and if the ions stop moving, that means the gradient will change. It will change. Number 10, which of the following statements about glucose, galactose, and fructose is most likely true? So what do we have here? We have isomers here, okay? The carbohydrates have different properties because they have different arrangements of carbon, hydrogen, and oxygen atoms. Okay, so they're basically like the same thing, okay? But it's arranged differently. Like they just have a different arrangement, but it's basically the same thing. If you like look at it and see the amount of OHs, the amount of Cs, the amount of Hs, whatever, like you'll get the same thing. And it's still C6H12O6, but it's just in different forms. Okay, and these different forms, they're called isomers. The isomers, they have the same chemical formula, C6H12O6, but they have different arrangement of atoms, so they have different properties. Okay, so which of the following statements best describe how organisms such as rabbits obtain the carbon necessary for building biological molecules? So we have a plant, we have grass with starch. Okay, starch is what? Starch is a carbohydrate. What do carbohydrates contain? They contain carbon. Look, carbon. It contains carbon. How does the rabbit get this carbon? The rabbit goes, it eats this, it gets the carbon. Okay. Rabbits eat plants and break down plant molecules to obtain carbon and other atoms that they rearrange into new carbon-containing molecules. So the rabbit will eat the plant will take the carbon and other molecules, and it will rearrange them in the way that its body needs. Which of the following observations was best suggest the alternative hypothesis? So what's the alternative hypothesis? Another group of scientists hypothesize that despite their biochemical differences, plant and fungal cell walls have similar functions. So which one supports that the plant and cell walls are similar? Obviously, that we're going to say that in both plant cells and fungal cells, the cell wall surrounds the outside of the cell membrane. We need a similarity between them, and that has a similarity between them. Okay, which of the following best describes the effect of a greater number of cysteine amino acids on the stability of proteins? Okay, so here we have a protein, okay, and what's interesting about these things called cysteine amino acids is that they have a disulfide bond. And this disulfide bond is very stable. It's a very stable type of bond. It's a covalent bond, okay? This disulfide bond is very stable. So if we increase the amount of cysteine amino acids, that means we're going to increase the amount of disulfide bonds. That means we're increasing the stability. Okay, so the change leads to increased protein stability because of an increased number of SS bonds in the tertiary structure of proteins. Okay, which of the following best describes a cell that is likely to have an extensive Golgi complex? Okay, so obviously if we're releasing digestive enzymes, we're going to need an extensive Golgi complex, which is why a stomach cell that releases the digestive enzymes will need an extensive Golgi complex. Why? Because the Golgi complex is responsible for modifying proteins, releasing enzymes, making glycoproteins, making glycolipids. But what's it called? It's mainly proteins, so digestive enzymes. Okay, proteins, enzymes, glycoproteins, and enzymes. Okay, that's like the main function of the Golgi body. Okay, so question number 15. Figure 1 represents a nucleic acid fragment that is made up of 4 nucleotides linked together. Okay. Which of the following characteristics of figure 1 best shows that the fragment is RNA and not DNA? Okay, so it's the identity of each nitrogenous base. Now, obviously, you can't tell which one is uracil, which one is thymine, which one is guanine, which one is cytosine. But just look at the options. Like the 5' to 3' orientation of the nucleotide. No, they both have 5' to 3' orientation, both the RNA and the DNA. The charges on the phosphate groups, شو دخل الcharges in identifying if it's DNA or RNA? Okay, and the type of bond linking the nucleotides together, it's the same bond. And there is no difference. The only thing is, we know that RNA has A connecting with U and C connecting with G, but DNA has A connecting with T and C connecting with G. Whatever, okay? Why is my handwriting so bad? Okay, so that's the only difference. What are A, C, T, G, U? They're nitrogenous bases. So the identity of each nitrogenous base is how we distinguish between the two, RNA and DNA. That's how we distinguish between them. Okay, so number 16, DNA and RNA are nucleic acids that store strong biological information, sequence of their nucleotide monomers, Which of the following best describes the structural difference between DNA and RNA? Okay, so what's it called? Here, let's go through the options. Okay, let's just go through the options. Okay, they say DNA contains 4 types of nitrogenous bases. Okay, whereas RNA only contains 2. No, it contains 4 as well. It contains A, U, C, G. DNA contains A, T, C, G. Okay. The backbone of DNA contains deoxyribose, whereas the backbone of RNA contains ribose. Obviously, that's true. Obviously, that's the main difference between them here. Okay, because they didn't like another main difference would be that the DNA is double helix, the RNA is like a single strand, but they don't mention it here. Okay, let's keep reading the other 2 options. So a DNA molecule is composed of 2 parallel strands. DNA is not parallel. DNA is anti-parallel. Okay, one of them goes from 5 prime to 3 prime. So one of them is from 5 prime to 3 prime, and one of them is from 3 prime to 5 prime. It's anti-parallel. Okay. So they don't have the same 5 prime to 3 prime directionality in DNA. Whereas RNA molecule is composed of only one 5 prime to 3 prime strand. That part of the answer is correct. Like RNA molecule is composed of only one 5 prime to 3 prime strand. That's correct. But the DNA is not only 5 prime to 3 prime. It doesn't have the same direction. They're anti-parallel. So one is 5 prime to 3 prime, one is 3 prime to 5 prime. Phosphate groups provide the rigidity to DNA, but RNA is flexible and contains no phosphate groups. No, just no. Okay, please. DNA contains 4 types of, the backbone of DNA contains deoxyribose, backbone of RNA contains ribose. That's the main difference between the two here. Which of the following statements best supports the claim that certain organelles within eukaryotic cells evolved from free-living prokaryotic cells? Endosymbiotic theory. What's the thing that backs this up? Some organelles contain their own DNA, like mitochondria and the chloroplast. They have their own DNA. That DNA is similar to the DNA that's found in prokaryotes. Okay, it's similar to them in structure and it's similar to it in function. Okay, so they say that a cell engulfed another cell and then the DNA is blah blah. They made up the endosymbiotic theory. Okay, so number 18. Oh, great, cell signaling. A researcher claims that epinephrine signaling pathway controls a catabolic process. Catabolic process is breaking down something, okay, in muscle cells. Which of the following statements best helps justify the researcher's claim? Okay, you see this big messed up drawing here? Let's make it very simple. They're asking us for a catabolic process, صح? Catabolic process, we know that it's a process that breaks something down. Okay, where do we see something being broken down into something else? Okay, so all of these downwards arrows, by the way, this is activating something that activates this, that activates this, that will activate that. Okay, it's activation. It's not breaking down. Okay, the downwards arrow is not breaking down. Okay, so the cyclic AMP, it activates this, that then activates this, that then activates this, and then the activation of this will cause this to break down into this, and the process of this breaking down into this because of this is called like glycolysis. Okay, just like hydrolysis is breaking a bond, glycolysis is breaking down glycogen into glucose 1-phosphate, and that is the only catabolic process we see in this big messy thing. Okay, which is why we're going to go with the glycogen phosphorylase catalyzing the breaking down of glycogen into glucose 1-phosphate. I think that's how they said it, no? Yeah, glycogen phosphorylase catalyzes the conversion of glycogen to glucose 1-phosphate. Which of the following best supports the claim that ethylene initiates the signal transduction pathway that leads to ripening of fruit? Okay, so let's read the question. Ethylene causes fruit to ripen. So ethylene is the thing that causes fruit to ripen. In a signaling pathway, the receptors activate the transcription factors, which ultimately leads to ripening. We want to see which supports the claim that ethylene is the thing that initiates the signal transduction pathway. Okay, we want ethylene to initiate the signal transduction pathway. Okay, so we want ethylene to be responsible for the ripening of the fruit. Okay, which one gives it to us here? Here it's answer D, because the loss of function mutations in ethylene receptors result in changes to the ripening process, meaning we mutated this ethylene. Okay, they're saying that ethylene underwent mutations. Okay, and then it lost its function. Okay, now ethylene lost its function. Because ethylene lost its function, this resulted in a change to the ripening process. So now the ripening process changed. What does that mean? That means that ethylene was the mastermind behind the ripening process. It was what was responsible for it. Ethylene was responsible for it. So when we lost the function of ethylene, okay, we then changed the ripening process. Something changed in the ripening process. Okay, let's go here and read the other options, see why they're wrong. Ethylene is a simple gaseous molecule which makes it easily detected by receptors. Obviously, that has nothing to do with anything. Okay, that doesn't mean that the ethylene is responsible for ripening. Fruit will ripen in closed containers without exposure to air. Okay, nice. How does that make ethylene responsible for it? That gives me nothing. Okay, then ethylene synthesis is under both positive and negative feedback regulation. That has nothing to do with anything. Okay, so look like ethylene synthesis is regulated by different feedback mechanisms, but this does not support the claim that ethylene triggers ripening through signal transduction. It has nothing to do with it. Okay, so you don't like get confused. I'm giving you the actual thing like to make sure. Okay, the reason is like just don't look at like positive and negative feedback and like say, oh yeah, this is 100% why. No, like don't look at the fancy words and say yes, this is the correct answer 100%. No, it has nothing to do with it. Okay, loss of function mutations, ethylene receptors, the mutations when they lose their functions, something changed in the ripening process. That's the correct answer. Okay. The molecular structure. Oh my god, how many times this come now analysis? Okay, so based on the molecular structure shown in the figure, which is most likely solid at room temperatures? Solid at room temperatures, the saturated fatty acids. We have saturated and unsaturated. Saturated, unsaturated fatty acids. Saturated, unsaturated. The saturated, no carbon double bonds. Unsaturated, there is carbon double bonds. Which one has the carbon double bonds here? Obviously, this is the one that has the carbon double bonds. This is the one that doesn't have the carbon double bonds. That means this is saturated, this is unsaturated. Okay, unsaturated, saturated. Unsaturated, saturated. Okay, the unsaturated one will be liquid, the saturated one will be solid. The saturated one is always solid at room temperature, the unsaturated one is always liquid at room temperature. Saturated, solid, both start with S. You can memorize it like that. Okay, so we're gonna pick the one with the saturated fatty acids, the palmitic acid. So the palmitic acid, because the absence of carbon-carbon double bonds allows the molecules to pack closely together. Absence of carbon-carbon double bonds means saturated. Okay. Which of the following statements describes a feature shared by mitochondria and the endoplasmic reticulum that increases the efficiency of their basic functions? Efficiency, we're talking about surface area. Surface area, usually there's folding involved. Both the mitochondria and the endoplasmic reticulum, guess what? They both fold. That means they both have highly folded membranes. That means they both have a high surface area. That means they both have high efficiency, because efficiency and surface area are interlinked. High efficiency means you're gonna have to have a high surface area. High surface area means that you're efficient. Okay. And surface area and folding are also linked. So a lot of folding, a lot of surface area. No folding, well, not no surface area, but like very small surface area. Number 22. Let's change the tab here. Oh my god. What that? Okay, yeah, 20. Where's 20? Okay. All eukaryotic cells contain at least one Golgi complex, typically located in the cytoplasm and near the endoplasmic reticulum. Which of the following best describes the process that occurs within the Golgi complex? We've said this 1,000 times. The function of the Golgi complex is the enzymatic modification of newly synthesized integral membrane proteins. Golgi complex takes the proteins, modifies them, makes them into what they have to be, glycoprotein, enzyme, takes the lipids, makes them into glycolipids, adds carbohydrate chains, folds them, packages them, sends them on their way. Okay, so it modifies and packages. So the process that occurs within the Golgi complex is the enzymatic modification of newly synthesized integral membrane proteins. Yani the modification of newly synthesized proteins. Which statement best explains the formation of hydrogen bond between the represented figure? So we know that a hydrogen bond is, from chemistry as well, it's a hydrogen atom with a phone, with one of these, with either fluorine, oxygen, or nitrogen. Obviously hydrogen is slightly positive, that means these are all slightly negative. So the definition of a hydrogen bond is a slightly positive hydrogen ion, a slightly positive hydrogen atom bonding to a slightly negative atom in its vicinity, not in the same molecule. This is not a hydrogen bond. This and this is not a hydrogen bond. I know that it's H with O, but it has to be in a different molecule. But this is water. Water is one molecule. It can't be in the same molecule. The hydrogen bond is between different molecules. Yani this is one water, and this is a whole different water. Okay? This is how a hydrogen bond forms. These aren't hydrogen bonds. Okay, this is a hydrogen bond. So the nitrogen has a partial negative charge. Here, look here, this is the hydrogen bond. The nitrogen has a slightly negative charge, obviously, because the hydrogen always has the slightly positive charge. And yeah, that's it. That's the answer. That's how the hydrogen bond forms. Which of the following outcomes will most likely result from a change in the shape of the mitochondrial inner membrane from a highly folded surface to smooth flat surface? If I take it from highly folded to flat, what will happen? What did we say? Highly folded means high surface area. High surface area means high efficiency. No folds means very low surface area. Very low surface area means very low efficiency. So the mitochondria will become less efficient because the surface area of the intermitochondrial membranes will decrease. Number 25, we did this also last time. So which of the following outcomes will most likely result from a loss of the ion pump function in the cell's lysosomes? We have lysosomes. Lysosomes are things that is the organelle that is responsible for digestion in the cell. Okay, so lysosomes are responsible for digestion. And the interesting thing about lysosomes is that they're very acidic. Okay, so their internal environment, the environment inside the lysosome itself is very acidic. What do we know increases acidity? H plus ions. This ion pump that they're talking about, it actually pumps out H plus ions and sends it directly to the lysosome. If we lose this pump, we're going to then lose the H plus. Then the lysosome will eventually become more basic, less acidic yani. More basic means more alkaline. They're the same meaning. More basic yani more alkaline. Okay? So the internal pH of the lysosomes will increase. The pH, as it increases, it becomes less acidic, more basic, more alkaline. As it decreases, it becomes more acidic, less basic, less alkaline. Okay? So the pH of the lysosome will increase because there's no more H plus ions. That means it's going to become less acidic, which will prevent the activation of the hydrolytic enzymes, because the only reason that the lysosome can actually digest things is because it has these enzymes inside it. It has these enzymes inside the lysosome. And the thing that activates these enzymes is the acidic environment. When the acidic environment goes away, the enzymes won't act anymore. And without these enzymes, we can't have digestion. Okay? It won't digest anything, which will prevent the activation of hydrolytic enzymes and interfere with the intercellular digestion of food. Okay? Number 26. Which of the following claims is scientifically accurate and consistent with an observation that a decrease in lysosome production within a cell leads to a decline in the mitochondrial activity? Lysosomes digest things into nutrients, so it gives off the nutrients. This nutrient is beneficial to the mitochondria. Okay, the mitochondria actually needs this nutrients to undergo cellular respiration, and what's it called? With fewer lysosomes there, it will be what's it called? With fewer lysosomes being produced, okay, there will be fewer lysosomes to actually break down the macromolecules and provide the necessary nutrients for the cellular respiration that the mitochondria undergoes. Okay, so that's the effect of decreasing the lysosome production. Less lysosome production means there will be less lysosomes actually available to break down the macromolecules to form the nutrients. With less nutrients, there will be less mitochondrial activity because there will not be as much nutrients for the mitochondria to actually undergo cellular respiration, which is the mitochondria's actual function. Okay, this question again from last periodic. Based on the model presented in figure 1, which of the following outcomes will most likely result from a loss of protein X function? Okay, so here we have protein X. Protein X went bye-bye. Okay, what was the function of protein X? The protein X was responsible for taking the very high K plus concentration and from taking the K plus ions from from inside the cell, which had a very high concentration actually, and taking it outside. The function of the X protein was helping the K plus ions, which were at a very high concentration inside the cell, to leak out. Okay, so we can balance the internal and external environments. But without it, the K plus will not leak out anymore. That means that the K plus concentration within the cell will then start to increase because it's not leaking out anymore. It's all inside the cell. For the concentration inside the cell will keep increasing. Look. Here the K plus that's outside is being pumped in. Look, it's being pumped in. This is the thing that was letting it out. Without it, it will keep everything in. معناته the concentration will increase. The membrane potential will be disrupted by an increase in K plus concentration inside the cell. Okay, so here they're asking us which of the following statements best describes how molecules released by the fish become nutrients for the plants. So actually these little cylinders are bacteria, okay, for your information. So here we have a fish. It releases this ammonia. The ammonia is then turned into nitrites, which is then turned into nitrates by bacteria. So the process of ammonia to nitrites and nitrites to nitrates are both done by bacteria, and then these nitrates are absorbed by the plant, and the plant uses them. Okay, so which of the following best describes how molecules released by the fish become nutrients for the plant? I just explained it. Let's see here. The ammonia molecules released by the fish converted by the bacteria to nitrates, which are used by the plants to make proteins and nucleic acids. Why proteins and nucleic acids? Nitrates have nitrogen. Nitrogen is a part of both nucleic acids and a part of proteins. Okay, it's a part of both of them. By the way, guys, make sure that you memorize the composition of each macromolecule. Like for example, carbs are made up of hydrogen, carbon, oxygen. Okay, carbon, oxygen, hydrogen. You need to know this because they're going to be asking you a lot about this, and some written questions as well. They actually require you to put the composition of each macromolecule. It helps to know this stuff. Okay, it's really beneficial. Make sure you know it. Okay, so the carbon is hydrogen, carbon, oxygen, lipid is the same, nucleic acid, blah blah blah, protein, blah blah blah. You have to know them. Okay, make sure that you know the compositions. They're in the course practice. You can ask ChatGPT, you can Google it, whatever. Based on the data in table 1, which of the following statements best supports the hypothesis that only certain types of molecules can move freely across the cell membranes? So as you can see here, researchers exposed identical cells to solutions containing different solutes. Each solution had a higher concentration of the solute outside the cells than inside the cells. So we had a higher concentration outside than inside. Solutes varied in size, charge, polarity. So the solutes that were outside the cell all varied in charge, size, polarity, whatever. Okay. After a set time, the researchers measured the concentration of each solute inside the cell to determine the permeability of the cell membranes to each solute. Okay, so here we have cells. Okay, each solute has each cell has a higher concentration of solute outside than inside. So we have a higher concentration of solute outside than inside in all three cases. Okay, how do I know that there are three cases, man? They're literally giving it to me, three cases here. Okay, whatever. What's it called? So yeah, okay. So solute A here was small and non-polar, small, neutral, and non-polar. Solute B here was large, charged, and non-polar, but it was large and charged. Okay, and solute C was small, but it was charged and it was polar. Okay, and what do we know about plasma membranes? The plasma membranes have the phospholipid bilayer. Okay, and the phospholipid bilayer is the interior of the phospholipid bilayer is non-polar. Okay, it's hydrophobic. So for things to pass by the bilayer smoothly, it actually needs to be similar to the interior of the bilayer. Okay, and to be similar to the interior of the bilayer, it has to be small, it has to be neutral, and it has to be non-polar. So anything that's small, neutral, and non-polar, meaning that it's small in size, it doesn't have a charge, and it's not polar, okay, it will move by very smoothly. Okay, so the correct answer is small non-polar solutes showed the greatest increase in internal concentration. Why? Because it moved from the area of high concentration to the area of low concentration smoothly, right through. It diffused through, no need for anything. على طول, easily through the bilayer. Okay, while the large non-polar solutes showed little to no increase. Okay, so if you're large, you can't go through as easily. If you're charged, you can't go through as easily. If you're polar, you can't go through as easily. You know, you can still go through, but you need help from what? From the channel proteins and from the carrier proteins. الله يسامحني على الرسم يا حبيبي. Like this, okay? And yeah, that's it. So, number 30. Which of the following observations best supports the model? So the endosymbiote theory proposes a model for the evolution of mitochondria according to the model. An ancestral eukaryote engulfed a small free-living prokaryotic organism. The engulfed prokaryote then formed an endosymbiotic relationship with the eukaryotic host. So what is the thing that actually best supports the model? The mitochondria and some prokaryotes share similar metabolic reactions that produce ATP. So we're backing up the endosymbiotic theory. The endosymbiotic theory is suggesting that something engulfed something. If something engulfed something, that means that the processes in both will be similar. And that's the only thing that shares similarities between them, which is why we choose it. Okay, so we go to number 31. Muscle cells have high ATP demands. Which of the following is a scientific claim about the structure of mitochondria and muscle cells? Should be different than it is in other cells for the high energy demands. Okay, so if we have high energy demands, we need them to be very efficient. How? Keep this in mind. So the inner membrane of the mitochondria and muscle cells should have more folds. Why? To increase the surface area. Why? Because the surface area and the efficiency are interlinked. We have high surface area, we have high efficiency. If we have a muscle cell, the muscle cell needs a lot of energy. And what produces energy? The mitochondria. And for the mitochondria to produce enough energy for the muscle cell, it has to be very efficient. And to be very efficient, it has to have a high surface area. And for it to have a high surface area, what does it have to be? It has to be highly folded. Which of the following best describes the structure of complex carbohydrates? You have to memorize it. So its monosaccharides are connected by covalent bonds. Okay. The monosaccharides are connected by covalent bonds. Here they're just asking you about the hydrophobic interactions. They're just asking you about the tertiary structure. Obviously, we know that the tertiary structure will have the hydrophobic parts inside, hiding away, and then the hydrophilic parts, the polar parts, the charged parts outside in water. Okay, because they like water. Here we have water, okay? The polar parts, the charged parts, the hydrophilic parts will be outside, welcoming the water. We wanna talk to you, whatever. And then the hydrophobic parts are inside. They're scared, they're shy, they don't wanna come out, so they just hide inside, okay? So these are the only correct two. I don't even know what these are. Like, that is an abomination, okay? This is something straight out of a movie, and that's a circle. Yeah, So no, we choose the first one. Which of the following is, if true about the drug, best supports the alternative hypothesis that the new drug will exhibit simple diffusion across the plasma membrane? So it's very easy. They already gave us the question in the question. So we don't have to read that big paragraph thing. They're asking us, they told us that the alternative hypothesis is, they told us that the alternative hypothesis exhibits simple diffusion. What exhibits simple diffusion? Small non-polar molecules. So we're going to choose that the drug is a small non-polar molecule. So it can actually exhibit simple diffusion. Which of the following conclusions is most clearly supported by the representation of nucleic acid 1 and nucleic acid 2? Nucleic acid 1 contains adenine-thymine base pairs, whereas nucleic acid 2 does not. What's it called? How do we know? How do we know? See here, this is a double strand. Okay, that means it's DNA. Oh my god, how am I this bad at drawing? Okay, that's DNA. Okay, they're telling us that this is uracil, this is thymine, this is cytosine, this is adenine, this is guanine. We know that DNA doesn't have uracil, and as you can see here, if you look very closely at all of these bands, no uracil is found. See, no uracil, 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 no uracil. So it matches the fact that it's a DNA, because DNA doesn't have uracil, it has thymine. Look, it has thymine. Look, here's a thymine, here's a thymine, here's a thymine, here's a thymine, here's a thymine. Okay, but it doesn't have uracil, which is good. That means we can say that this is a DNA. That's right. We draw the double helix, we're correct. Now, in this one, we're also correct because they only drew a single helix. And look here, they included uracil. Here's a uracil, there's a uracil, 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 uracil. There's no thymine. As you can see here, there is no thymine. No thymine means that we can draw a single strand and we can call it RNA. Okay? So adenine, thymine for nucleic acid 1, nucleic acid 2, no adenine, thymine. Okay? Oh my god. Okay, question number 36. Come on. Okay. The observed differences between... Okay, it's multiple choice. Okay. The observed differences between the groups most likely resulted from differences in the ability by the seedling to produce which of the following monomers? Okay. So here they're telling us that the seedlings in the potting soil, the height was 18.5, mashallah, and the leaves have a dark green color and are normal in size. Okay. Here we have in the sawdust 4.8, quite sad, and then the leaves have a grayish color and are small in size. Okay. And what do we know is responsible for height and like growth? Proteins. Okay, and what's so special about the composition of proteins? It's that they're composed of nitrogen. Okay, what else is composed of nitrogen? Nucleotides as well. Okay, DNA, RNA. Okay, so if we have a shortage in proteins, that means we have a shortage in nitrogen. That means we also have a shortage in nucleotides. How do we know that there's a shortage in protein? Because look at its height, for the love of God. Okay, look at the height. It's 4.8 centimeters. Here it's 18.5. This is way less than this. Okay, and height is like managed by proteins. Like growth is managed by proteins. Okay, so the observed differences between the groups most likely resulted from differences in the ability of the seedlings to produce which of the following monomers? Amino acids, nucleotides. Amino acids, the monomer of proteins, because they're asking us for monomers. Amino acids, the monomer of proteins, and nucleotides, the amino acid of nucleic acids, the monomer of nucleic acids. Okay. Number 37. Which of the following is the accurate description of the process shown in figure 1? Here we have a dehydration reaction. How do we know that we have a dehydration reaction? We have a bond being formed. Excuse my circling. We have a bond being formed, making a big, a big molecule. And we have water being released. Water being released, dehydration condensation reaction. Okay, dehydration condensation mean the exact same thing. Formation of a covalent peptide bond in a dehydration synthesis reaction. How do we know that it's a covalent peptide bond? Look, You have one amino acid and one amino acid. How do you know that it's an amino acid? You have an R group, you have an amino group, you have a carboxyl group. Equals amino acid. ما شاء الله على الخط يا أخي حبيبي. Okay. Number 38, for the love of God, just enough. Which of the following best describes the formation of the bond shown in figure 1? Again, a covalent bond is formed between a carbon atom and a nitrogen atom along with the formation of H2O. That's how everything happens. Formation of H2O, synthesis reaction, dehydration reaction, condensation reaction, they all mean the same thing. It's just different names for the same reaction, okay? It makes a bond between two elements of two different molecules, releases water, okay? So here the bond is between carbon and nitrogen, okay? That's the bond that it created, and it released these two hideous things. It actually released this one and this one, but same thing, okay? And yeah, that's it. It released H2O. 39. Which of the following best describes the numbered areas? Okay, so obviously number 1 and number 3 are hydrophilic because it is in the region where water is, okay? It's the hydrophilic heads. So obviously it's hydrophilic. And it's polar because being hydrophilic is also, you're also probably going to be polar, okay? And we know that the hydrophilic heads of the phospholipid bilayer are also polar, so region 1 and region 3 are polar. And then region 2 is in the middle of the bilayer, which is where the hydrophobic fatty acid tails are, which are also non-polar, okay? But the correct option is only talking about 1 and 3. That's all you have to actually identify, so you're good for this. Number 40. So here they're asking us which of the following membrane components are most likely non-polar. What do we know that is non-polar? That anything that's non-polar has to be embedded within the membrane itself. It has to be in this region, okay? Now you're going to tell me طيب ما the channel protein is embedded, ما this glycoprotein is embedded, this integral membrane protein is embedded. Yes, they are embedded, but they're partially embedded. Half of it is inside, half of it is outside, half of it is inside, half of it is outside. They want only non-polar. They're not asking you polar / non-polar. No, they're asking you only non-polar, okay? What is only inside the membrane? The only thing that is like purely inside the membrane is this cholesterol, okay? And the tails themselves, okay? And by the way, usually in diagrams, most of the time they draw cholesterol like this, like a lightning bolt, okay? So you can like identify it quickly. Okay, so based on the figure, which of the following statements best explains why the nuclei sample had a higher level of radioactivity than any of the rinse water samples? Let's go up here and see. Okay, so basically they're taking a cell, okay? Here they are taking a cell, and they're adding this radioactive phosphorus to it, okay? They're adding this radioactive phosphorus to it so they can actually detect it, because you can't detect regular phosphorus, it's very hard to detect. So you'll just add radioactive phosphorus, it's way easier to detect because it actually gives off radiation, so your life becomes easier as like an experimental scientist, okay? So they added the radioactive phosphorus to this cell, and then they actually took out the nucleus of the cell as well. So they extracted the nucleus out of the cell, and then they started to put water on it. They put water on it once to get rid of the excess phosphorus, because they want to see only the nucleus. They want to see only the radioactivity of the nucleus. They don't want to see they don't want to see the excess radioactivity, the excess phosphorus that's outside the nucleus. So they rinse it with water. They rinse it with water once, twice, 3 times. All the excess radioactivity is gone. Okay. All the excess radioactivity is gone. And by the way, sorry, they don't rinse the nucleus. They rinse the cell first. So they rinse the cell 3 times first. So they rinse it 1 time, 2 times, 3 times, and then they extract the nucleus. Okay, then they extract the nucleus. And what happened was, So you see, the darker and bigger it is, the more radioactive it was. So what happened is that these are the rinsed water samples, okay, and this is the nucleus sample when it got extracted. See, notice how the rinsed water samples are less radioactive than the nucleus. That means that phosphorus was used in the building of nucleic acids in the nucleus, because the phosphorus is radioactive. So if the nucleus is the most radioactive thing between these four, okay, we're not counting this because it had a higher level of radioactivity than any of the rinse water samples. These are the rinse water samples, okay? These are the rinse water samples, ماشي? And this is the nucleus. Okay, so why did the nucleus have a high radioactivity? Because the phosphorus was actually used in building nucleic acids inside the nucleus. Okay, so the nucleus took all the phosphorus that it could find and started to use it. Okay, that's why it was more radioactive, because when you're using the radioactive material, you're going to become radioactive, and you're taking in the phosphorus that's radioactive, you're going to become more radioactive. Okay, that's the whole thing. I solved this last time. Basically all it is, they're telling you that for every 5 kilometer increase in the distance from the shore, the radius of the cell decreases by 50 micrometers. So if the radius, if the diameter, the diameter decreases by every 50 micrometers for every 5 kilometer increase in the distance, okay? So if the diameter was 900 micrometers, what would happen if I move 25 kilometers? So for every 5 kilometers, there's a 50 micrometer decrease. So for 25 kilometers, it will be 25 times 50 divided by 5, which is 250. Okay, so there will be a 250 decrease. So here there was a 50 decrease, here's the 250 decrease. The original was 900, so 900 minus 250 equals you to 650. Next question. Okay, so which of the following best explains the observations represented in figure 1? Okay, so the cell in the pond, which is isotonic, okay, nothing happened, okay, so because like the pond isn't pure water, okay, so it's like a little bit of water, a little bit of this, like it's isotonic. It's the concentration of water outside the cell is the same as inside the cell, okay, so it's isotonic, so nothing will happen, okay. Here the concentration of water inside the cell is more than the concentration of the water outside the cell. How do we know? The cell is in a sugar solution. أكيد the sugar solution will have less water potential. معناته the water will go from inside the cell to outside the cell to balance the water potential to achieve equilibrium. So the water goes from the inside to the outside. So our cell starts to shrivel. Okay. In this one, our cell began to expand. In the last one, our cell began to expand. That means that the distilled water had more water potential than the inside of the cell, meaning it's called hypotonic. And here in the second case, it's called hypertonic. Okay, so when the water potential outside is more than the water potential inside, it's called a hypotonic solution. When the water potential inside is higher than the water potential outside, it is called a hypertonic solution. Okay, and that's it. So look here, there was a net movement of water out of the cell suspended in the sugar solution. There was a net movement of water into the cell suspended in distilled water. Just like I said, which of the following statements best describes the formation of complex carbohydrates? It's a repeat of what we said. Which of the following best describes the structures of carbohydrates? They occur as monomers, chains of monomers, and branch structures. There is no easy thing, there's nothing for me to say here. Just memorize it. Okay. Here we are seeing the opposite of a dehydration condensation reaction. We are looking at hydrolysis. Hydrolysis is when you add water to a big molecule to break it up into two smaller molecules. Okay, that is what's happening here. So it represents a polypeptide chain that is broken down through a hydrolysis reaction. Okay, 47, please, anytime. Okay. So which procedure should be done next to gather the data needed to meet the scientist objective? Okay, so basically in this experiment, they're trying to find the maximal rate of transport of this protein. Okay, so they're looking at environments, okay, for the specific cell that will maximize the rate of exchange. Okay, the rate of exchange at 25 degrees Celsius was maximized at this concentration. Was maximized at the concentration of 1. Okay, so we found the optimal concentration. Okay, طيب, but we know that the temperature also plays a role in transport rates. We know that. طيب, we have to find the optimal temperature as well. So we have to test. We keep our optimal concentration. Okay, we're going to keep our optimal concentration. We're going to use it, and we're going to see at which temperature does this one concentration make the biggest transport rate. Okay, so at which temperature is the transport rate maximized at a concentration of 1 micrometer per milliliter? Okay, so we incubate samples containing 1 micrometer per milliliter of ATP at 4 temperatures other than 25 degrees Celsius. So we start trying different temperatures. Which of the following predicts the type of cellular defect that is the most likely cause of the researcher's observation? So a researcher is studying a specific type of cellular defect in animal cells. She notices that cells will have a defect, with this defect have reduced energy storage, okay, and decreased cell membrane stability. What is responsible for energy storage? Long-term energy storage, fats. What is responsible for the cell membrane? Phospholipids. Okay, so it has what's it called? So the defect in the fatty so it has So the cause of this defect is a defect in fatty acid synthesis, because when we reduce fatty acid synthesis, we have reduced formation of fats and reduced formation of phospholipids, because both phospholipids and fats need fatty acids. Okay, so a defect in the fatty acid production will cause us to have a decreased formation of fats. That means decreased energy storage, and decreased amount of phospholipids. That means decreased stability in our cell membrane, because our cell membrane is made of what? Phospholipids. Which statement correctly indicates the cell that is able to more efficiently exchange materials of the external environment? So they told us that it's a spherical bacterial cell, so it's a sphere. We're talking about a sphere here. Okay, they gave us the radiuses of both the bacterial cell and the human egg cell. Okay, what they want us to do is see which one is more efficient. So we have to find the surface area to volume ratio. Okay, we have to divide the surface area by the volume. The surface area, you find it by 4 pi r squared, and the volume you find it by 4/3 pi r cubed. Okay, you find the answer for each, so you substitute 3, then you substitute 100 to find the surface area of both, and then you substitute 3 and you substitute 100 to find the volume of both. After you find that, you divide the surface area by the volume of the bacteria. So you substitute 3 micrometers into the surface area, 3 micrometers into the volume, and then you divide the answer of the surface area by the volume. You do the exact same process for the human egg cell. The one that gives you the bigger answer is the one that is more efficient. Here it is the bacterial cell, I think. Yeah, the bacterial cell, because it has the largest surface area to volume ratio. It had the bigger answer at the end. Which of the following best describes how the properties of water at an air-water interface enable an insect to walk on the water's surface? The thing that enables insects to walk on water's surface is actually called high surface tension, and the high surface tension is made possible by hydrogen bonds. Okay, so hydrogen bonds between molecules at the surface of the water provide surface tension. Same thing here. Which of the following cells represented in the figure, of the two cells represented in the figure, which would likely be more efficient at exchanging substances with the surrounding environment? Again, surface area to volume ratio for both. You find the surface area of this divided by the volume of this, you get an answer. Same process. You find the surface area of this divided by the volume of this, you get an answer. The one with the bigger answer is the winner. The way you find the surface area of a the way you find the surface area for this shape is you find the area of this side, multiply it by 2. You find the area of this side, multiply it by 2. You find the area of this side, you multiply it by 2. How do you find the area of a rectangle? Length times width, okay? So find the area of this side, multiply it by 2. Find the area of this side, multiply it by 2. Find the area of this side, multiply it by 2. Then add all of the numbers together, you get the full surface area. As for the volume, you just do length times width times height. Okay. As for the cylinder, I genuinely just forgot the form. Like, I solved it last time. I solved it in the last quiz. I gave you guys the formulas in the last quiz, but I forgot the formula right now. I know that you find the volume of a sphere by pi r squared h. I forgot how you find the surface area, so just look that up. Okay, ChatGPT, Google, whatever, and find that the surface area to volume ratio, the one with the bigger surface area to volume ratio, is your more efficient cell. Okay. Number 52. Which of the following best predicts the effect of greater proportion of saturated fatty acids as compared with unsaturated fatty acids on the stability of the membrane? Okay, so obviously the more saturated fatty acids, the more the membrane will be stable, and that's only the more fatty acids we have, the more stable our cell membrane will be. Why? Because these fatty acids that are saturated pack very closely together. They can pack very closely together, but the unsaturated ones are farther apart. Okay, so the closer you are, the more stable you are. That's why the saturated is more stable. Okay, yalla, come on. Okay, here, based on figure 1, which of the following experiments would best confirm that the protein A utilizes passive transport while proteins B and C do not? Obviously, we need to we need to see how everything is consuming ATP, because the thing that actually tells us if it's doing active transport or passive transport is the ATP production. If we have is the ATP consumption. If we're consuming ATP, that means it's active transport. If we don't consume ATP, that means it's passive transport. Okay, so it doesn't consume ATP, consumes ATP, it's active. Consumes ATP, ya Allah. Consumes ATP is active, does not consume ATP is passive. Okay, what's it called? We just compare the ATP consumption of cells expressing each protein type during the solute transport. Okay, so we just see how much ATP it actually consumes, and from there we can tell if it undergoes passive or active transport. Okay, based on the functions of macrophages and B lymphocytes, which of the following claims is most likely true of the organelles needed in both types of cells? Okay, so the B lymphocytes and the macrophages, they both help in fighting infections. Okay, the macrophages actually engulf bacteria, like they digest it, they destroy it, and then the B lymphocytes actually produces a protein that sends it sends to bind to the bacteria and then neutralize it. Okay, so what actually digests things? It's enzymes. What creates enzymes? Golgi complex. What's it called? What creates proteins? What creates proteins? The endoplasmic reticulum. And what do the endoplasmic reticulum and the Golgi complex have in common? They both transport proteins, صح? So they need organelles that process and transport proteins. Okay? Is that it? Yes, that's it. Oh my god, thank god. Okay, so those are the MCQs. I will try and post the FRQs as soon as possible. Okay? For now, go ahead, study the MCQs. Okay, make sure you're ready for the final, guys. Please do the course revision, do the AP classroom, this one, the one before that. All 3 videos are out. Watch all of them. Okay, not even watch, just know them. Okay, if you watch me or you don't, it doesn't matter. You guys have to study really hard for this final, because it's 50% of the grade. Even if your bio average is 100, okay, because there are some people with very high bio averages, okay. Even if your bio average is very high, if you don't study for this exam and you go and you mess up, your average is going to get really messed up. Okay? So be careful, do this. Go study the course revision, then go study the course practice, then go watch my videos. At the end, you watch my videos. Okay? You go do the course revision, do the course practice, then you go watch my videos. If you need any help, if you want any extra video, if you want me to make a summary or something, whatever you guys need, just somebody text me on the bio group, text me privately, whatever. Just let me know so I can actually do it, because I'm not psychic. I don't know what you guys want. So just let me know, okay? Thank you guys. Have a nice day, and good luck to genuinely everyone.